So what I thought I'd go over, if we take turns trying to educate on stuff, some of this actually was just in Tyler yesterday, and there are about 40 of their people over there where I was going over targeted therapeutics development. And we were talking about clonal pressure, and we know a lot more here, but it never hurts to sort of refresh our memory of these things and sort of look at examples of what we've experienced and get updated on almost what we presented before. So that's a little bit of what this is. Some of this is stuff you guys have heard before, but it's sort of newer thought. When we get other people to work with us, we'll share some of this information. We'll see what their input is, and so we modify things to that. So this is sort of an upgraded version of Target the Target, but specifically some of the work we've been doing in Ewing Sarcoma, mainly to tie in with Tyler here. Who's played a role with Tyler? Everyone? Ed has, yeah. So, and he's a unique personality, but he's a nice kid. But so anyway, so we'll touch on that stuff too. So I think the whole original reason for setting our clinic up was to basically, well, it wasn't right in the beginning. It was to do targeted therapeutics, but then we started to figure out, there's different kinds of targets. Some are your gene signals, but there are immune targets, and we're learning them more and more. And it's kind of a left-out child, but it may even be more important than the signaling targets as we go along, because there are broader immune applications, and the immune field seems to be advancing faster than the molecular targeting field in a sense because of the checkpoint inhibitor technologies. So typically, what we're now engaged in, it was really after Allison, Cason's, the young girl with the Southwest Air napkin that we wrote on. Do you remember that? You hear about that story? Never heard that story? So Dave Shannon and I were over visiting her. She had colorectal cancer, trained to be an Olympic swimmer, but then developed cancer, kept swimming throughout her chemotherapy regimen. She lived in Missouri, St. Louis, and had a boyfriend and everything. Was always doing stuff, real smart kid. And anyways, couldn't keep up the swimming through the FOLFOX porphyry, but worse, she developed an infectious abscess on like cycle 4 or 5 of her FOLFOX regimen, and the surgeons just decided that she's so far along with her cancer that it's not worth trying to clear out the abscess, so we should just let her die. So her mom had talked to me about possible approaches, and we had lined up maybe a possible study, but she had to stabilize after the surgery. But then when the surgeon felt that the abscess wasn't worth going into, I actually talked to Joe Kuhn, and Joe said, hey, transfer over here. And he cleared out the abscess, the infection completely cleared, and she lived about another year after that. So we did try to design some trials, but that just shows in some of the real world, if you have cancer, if you have metastatic disease, and she was, I think, 24. Wow. So she was too far along to try to pursue any further clear out an abscess. Simple procedure. But thanks to Joe, she was saved at least for that extra year. But the point is, while we were going back and forth, and we started to realize that, okay, maybe we bought an extra year with antibiotics and the abscess clean out, what could we do? So I had always kept my eye on the fields and how they're looking at targeted approaches. The RNA interference came up as a very attractive means of controlling any DNA expression. You can construct an RNA interference signal to any DNA signal. And so the thought was, if we could create a technology that would quickly hit the cancer to a specific molecular signal, that's great, but there's a lot of molecular signals cancer cells have. So if you hit one, there's like another that's going to crawl out. And then you hit that one, then another one's going to crawl out. It's like that game in the circus, you know what I mean? So that's not our whole solution. So what we would need to do is hit the cancer fast, get it specific so you don't have side effects. So it'd be targeting cancer mutations makes a lot of sense. But then try to prevent recurrence or delay recurrence by activating the immune system. So that's where the 1-2 punch idea came out. And it was basically from a discussion that Dave Shannon and I had a plane coming back from visiting Carly if she had come here, and we got to know her here, and we just went over there to visit her. And while we were on the Southwest Air plane, he was just totally frustrated. And I always have ideas, and I always know medical things, stuff like that. But it's totally frustrated. So I tried to explain to him real simply. And so I took a Southwest Air. He goes, if you had a whiteboard, what would you do? And I go, well, I got a white napkin. How's that? And so we wrote it a Southwestern napkin. He's got it in a safe, and that's how we decided to. Yeah, I'm not kidding. So that's how we brought in Donald Rao into Gridalis and Strike Stuff to form that company to develop the RNA interference technology, and that became the direction that we started to then move at Crowley. That's why we had to know what the gene signals are. Why are they? What therapies targeted? And what companies can we bring these signals in from? That's why we set up programs to bring in the targeted therapeutics and immune therapeutics, to try to get a 1-2 punch. And so what's happened now over the years, I never wanted to get into pediatric management, but in Ewing Sarcoma, we now have the bifunctional RNA interference lipoplex that's targeting the signal, the clear indicated signal causing Ewing sarcoma, the USFLI protein, and we have the immunoapproach targeting the immune mechanism. And so we now have the two actual arms to do the 1-2 punch with from kind of like our own creation as a big team. So that's why I picked that as sort of a topic. Does that make sense? That's the rationale on it. So two directions. And so on slide 3, in very simple terms, there are gene signals that control cancer cell survival, any cell survival, cancer, any cell growth, and spread. What happens is that our normal cells have normal checkpoint regulations for everything they do, when they grow, when they metabolize, when they get rid of waste. And so when a critical mutation occurs in a normal cell, if it's involving one of those signal controls, well, then you're going to mess it up. You'll either make it worse or you make it better. And if you make it worse, and that cell performs less optimally than a normal cell, what do you think it does? Rachel?
Why are you looking at me?
Because you're always a morbid person. It's the perfect question for you. And
can you ask the question again?
Okay, hint, it dies.
It dies.
Yeah, that's to you. So I mean, I'm assuming every answer you have is it dies, or I lose my head, or I lose my leg. So I'm trying to give you help here. I'm trying to make you look smart, and even so.
I was thinking about my amputated foot. That's
what I'm trying to get you to focus. If you're a smart person, if you focus, we'd really get good answers out of you.
Okay.
Like
I'm ready. Bring it.
Point is,
okay.
Yeah, I did. So point is, if there's a signal in a cell, and all these cells are competing with each other, but they all have even signals, they're all going to grow happily right next to each other. You can eat as much as the next one, you waste as much as the next one, you find that environment, you grow happy together, okay? Happy, everyone's happy. However, if one of the cells gets a more aggressive signal, okay, to grow more rapidly, well, he's going to make 2, 3, 4, he's going to compete better in that region, he's going to grow up. That's what a cancer cell does. And so that cell that has a mutation that gives an advantage in growth or spread is going to start to take over eventually, okay? Now, your immune system figures this out, and they're going to try to keep it down. And they'll keep it down for years, like 20, 30 years. We all have cancer in our bodies right now, But our immune system's keeping it down, or it's eliminating it. So, on the opposite side, where I tried to help Rachel, if that signal is the opposite, if it's a deteriorated signal, the cell can't compete. The normal cells will grow over, and that cell dies. And that's your apoptotic function, okay? 99% when you get a gene mutation, probably even more than that. That's scary.
I don't know.
Yeah. Okay. This is difficult here. Okay. Great head show. Rachel focused on death. So what's next? So then if you have that, 99.9% of gene mutations that occur in cells are naturally handled by the p53, they undergo apoptosis, and we never know about it. Our body's naturally regulating that stuff. So a lot of mutations, most mutations are nothing. They're defective. But those rare mutations that add in an advantage will give that cell a competitive edge. But if it doesn't know how to spread, it'll grow up more and more and more there, and maybe it'll become a wart. Well, that's not so bad. But if that wart cell can figure out how to spread, well, it's going to be a pretty mean wart if it starts getting all over the place. Maybe it'll be lipomas then. Well, then if it gets another mutation, then it's now something pretty aggressive if it doesn't have the mortality signals that it normally would have. So that's what goes down to genetic signaling control. So we got to figure out what are those signals that are important for cancer cells to grow to have an advantage, okay? And we know what a lot of those are. And so that's what we try to target. So on this slide here, this is from Neil, these are the 12 key critical pathways. When we try to figure out what are the pathways that would give a cell an advantage over, one cell over another cell, in essence, a cancer cell over a normal cell, these pathways, which deal with genome maintenance, that's where the P53 comes in, cell fate or cell survival. Cell fate means if it gets to other areas, it can survive in the blood and stuff like that, or progress in. So these are the genetic signaling pathways. It's not just one gene. One gene is messed up, but that gene is messed up in a pathway that has a control. Every function in a cell is like a series of switches. You have a switch with a light. You flip it up, light goes on. It's a series of little copper ions right next to the copper wire that go all the way to the light bulb, and then it turns on. But if you disrupt one of those and break the wire, it's not going to work. And so that's how a signal pathway works. And that's how it works genetically. It's just as simple as that. So if we find the right gene to target, it doesn't have to be the gene that's necessarily mutated. It could be downstream from that. We block that one, and the mutated one won't work. Okay, can you prepare yourself?
Okay, I'm ready.
If we have a mutated gene, and we block downstream, we stop that mutated gene function that's always on, right?
Mm-hmm.
And so that cures it. Now, if we have a mutated gene, and we put a therapy in upstream, what would we do to the function of that mutated gene? Would it block it?
Yeah. No, because it would be going this way.
Right.
So it would keep going.
Right. You are right. Good. Excellent. Perfect.
Redemption.
Yes. You get a 50% on this test. That's great. But you'd be amazed at how many companies have come up with products targeting signals that are upstream from the mutation. And I'm like, well, I hate to say it, but EGFR blocked with RAS mutations. EGFR is above RAS. Now, it does have some other roles around RAS. We still may get some activity, but technically, the main proliferative function of EGFR stimulation when a mutation there is through RAS. So if you have a RAS mutation and you're blocking EGFR, you're very unlikely to get a good response. You might, but that'd be sort of in a Bristol study, but technically, there's a lot behind that. So but the point is who at any company would develop something and spend $1 billion for it, knowing that? And the problem is they don't know it. Okay, what?
What? We're just recording it. It's fine.
Right, good. Right. Anyways, you get what I'm saying. So I'm trying to explain how important it is to just know the simple thing of a light switch turning on a light, gene signal pathways like that. So we got to get that. All right. So the next slide, this is important. I don't want to challenge you with this one.
No, come on. I'm ready.
So if you have normal cells, that would be the yellow ones, okay? They all have normal genes. And cancer is like a population of people. In a nodule the size of a pea, there are 10 to the 6 cancer cells. In a nodule the size of a marble, there are 1 trillion cancer cells. So guess what? Are they really all identical? Okay, that's too easy. So even though you got it, I saw the head shaking. So they're not, but they're not. And so when you treat a cancer, we see the mass shrink up and down with a CT scan. And that's great when it goes down, it's bad when it goes up. But inside the cancer, your therapy is getting to each individual cancer cell, okay? And if it's sensitive, it'll die. If it's not sensitive, it'll be resistant and it'll survive. And so the shrink will be dependent on how sensitive the cancers are. Well, with chemotherapy, it's broadly across any genetic pattern. It's kind of like if you have a bunch of people in a conference room and you put in bad-smelling air, everyone's going to smell it. And it may be enough to kill a few people, but most of the time you're just going to be suppressed for a while and you get out of the room. Okay? I got to say it's original. So it's general is what I'm trying to tell you across the board. And if you get a response to chemotherapy and you have a genetic signal that's before chemotherapy and you get a response and you test the chemotherapy later, it's going to be the same mix of molecular signals. It's possible to have some changes, randomly, but for the most part, it's going to be not much different from patient to patient to patient, although because cancer tries to recover from chemo, it'll sometimes change its profile because there's more mutations, they're dividing more, so you can have some more increased mutations during the chemo phase, but typically it's over a period of years, not over months with a response and no response. Now, if you have a targeted therapy, and the targeted therapy to a particular gene really works, you're going to be harming all those cells that have that gene signal. And so if it's this pink, blue, green one, and it's against the green molecular sensitivity signal, if it's really working, it should kill all those pink cells. But you may get it down to less than 10 to the 6 cells, less than a million cells. And if we get the cancer down to less than a million cells, then what kind of response will we call that?
Like a partial response?
Complete response.
Complete.
Because below 10 to the 6, you can't see it by PET scan.
Fun fact.
You get it?
Yes.
But even though it's a complete response, the cancer's still there. Yeah. And so, does the patient with a complete response have a risk of recurrent disease? And that's why. So now, if we've done that and it's gotten a complete response, and the cancer recurs, then of the pink and the blue and the green, it's tough. You're going to get a good education today. So
I'm not sitting by you again.
So you're right here. It's so easy. The pink has this one genetic profile that was sensitive, and the therapy we gave was attacking the sensitive profile. But these guys survived because they didn't have sensitivity. So if the pink all goes away, and the mastering done, we can't see it. But now we start seeing a new mass coming back like 8 months later. What color do you think this new mass would likely be?
It'd be blue and green.
Blue and green. Correct.
So then you do a molecular profile on that to see which ones are driving that new cancer. Yes.
That is the Mary Crowley response. Excellent. Yes. Everyone on the phone needs to hear what Rachel just said. But you're right. But now you get it, and you'd be shocked at how many people don't get that. And so this was actually developed by AstraZeneca without AstraZeneca knowing it. And I'll explain why. This is sad. So in 2005, a group of scientists got together, and they realized that EGFR was an important signal. They developed EGFR small molecule, EGFR antibodies, and they showed that they could kill cancer cells, lung cancer, but a variety of cancers, but lung cancer with an EGFR receptor inhibitor, EGFR TKI. And they went to Bristol. They said, hey, look, we discovered this. And Bristol says, great, perfect treatment for lung cancer. So they treated, a ton of lung cancer patients, and they showed that the overall response, the overall survival was 4.7 months in second-line patients second or third-line patients who had the EGFR, 6.7 months who had EGFR TKI, and 4.7 months with chemotherapy. I think it was single-agent taxes here. And so that was statistically better. However, if you're just sitting here and you say, oh, wow, I can live another 2 months. I mean, seriously, is that really what we want to do with cancer? So it achieved statistical significance. It was like a 1,500-patient trial. And that's great. They got their product approved. But someone else came up 4 years later and said, well, if we look at, this is really designed to kill the EGFR. It's an EGFR receptor inhibitor. If you had an EGFR mutation, wouldn't it be better in those patients? Wouldn't you think that? And Bristol said, no. Well, what if we just look at it? And so finally someone looked at the database and said, well, let's look at the patients who had the EGFR receptor mutation. And I think 13% of these patients had the mutation, the rest didn't. And I said, well, what's the survival? They all got the EGFR receptor, inhibitor. What's the survival if you had the mutation or didn't have the mutation? And then, or not the survival, they did look at that, but the overall response rate. So the overall response rate overall at first was 8.9%, and then when they looked at it, but only looked at the targeting, it was 70%. 70% of these patients who had the EGFR receptor mutation actually had a rhesus PR or CR, as opposed to the mixed group of 8.9%. So it's even worse than I thought. I didn't notice that when I first put this on the slide. So point is, they were having trouble moving it into frontline therapy, and the pressure was to get into frontline therapy, but it wasn't good enough if you used the whole group. So that's why they started to look at these other signals. Now, you'd like to hope you look at the other signals because it's the best advantage to the patient. You want to match up the right patient with the right trial. So sort of that happened. but it allowed them to get into a revenue plus situation rather than revenue negative if they did the right thing. So, okay, we're capitalistic, but you get why Crowley exists now. So that's true, it's sad. So then when they looked at it in frontline, but they had it as only the EGFR mutated patients get therapy, and the only EGFR mutated patients get frontline therapy, chemotherapy. When they looked at that, then they had the response rate 65% versus 16%, and they got it approved. Okay, so to me, they proved that matching the molecular signal with the therapy makes sense. So then a group came up and they said, well, what does happen with these patients who relapse after having this clonal therapy? And the smart guys were kind of talking to business guys who were counting the dollars, but the smart guys, they said, you may want to look at the molecular profile of those patients who actually relapse after they get a good response to an EGFR receptor inhibitor. And so they started to do that at AstraZeneca. This is actually about when I came in and got involved with them and actually looked at the database right when they had discovered the T790 mutation. And I'm like, well, dude, that seems like a good target. Why don't you use that as a small molecule activator and look for what can knock down those cells with that mutation? They go, okay, great. So then they found that, and they said, okay, well, we're going to do another study in relapsed patients, and we're going to treat the relapsed patients with EGFR, in this case, it was Tagrisso, with the second generation EGFR inhibitor. And so they're about to start the study. I'm like, wait. But why are you targeting the MET amplified one with that? Why are you targeting the HER2 amplified one? Did your cell lines say that they're sensitive? And they went back and looked at all their cell lines, and the T790 mutated cell lines all were dying, but the HER2 amplified cell line, the MAPK1 amplified cell line, the PI3 kinase cell line, they're happy. It didn't bother them at all. So well, why would you do that? Well, let's just up the power of the study. Let's just up to 1,000 patients. No, wait, why don't you just downsize the study to only include the T790 mutated patients? Why would you up it? Oh, yeah, we could do that. You would be shocked at how it was a room of like 20 people, and I'm like thinking like I'm an idiot. Like, guys, you don't see this? No, this really happened. And so they downsized it, and it was positive, and now Tagrisso is approved by FDA. And so it's amazing, but that's the first prototype example of matching the target with the targeted therapy. And if we can understand this, it seems simple, like turning on a light with a light switch and cutting the wire in between, but that's a hard concept. We gotta try real hard to think about it, understand it, ask questions, ask me, ask Neil, and ask Rachel now. So you get what I'm saying. So it went on and got approved. Other examples of targeted therapeutics, the HER2 is the first one, but it's a little bit different in that it's a binder to a conjugate, but it's the first one because they're targeted therapy. But the real targets we're talking about are when there's a molecular signal and you can disrupt that signal. You can identify a patient with that target and block that target. And that's where some of the fusion proteins come in, EGFR mutated proteins, as we already talked about, crizotinib with rearrangements, crizotinib with the ALK arrangements. And you can see all these dramatic responses of patients with bulky disease. And some of these response rates are in the 70%, 80% range. They could be third, fourth line patients. I mean, we've had, there's the experience of ALK mutations not being known, patients going front line, second and third line, prognosis 3 months to live. Oh, ALK mutations identified, put them on crizotinib. Whoa, 85% response rate. I mean, so if you can really match the right patient up, you can turn the tide on what the cancer is basically taking your body to. Even with those discoveries, it still took years before basically grants started to prove studies along those lines. And what it took was big pharma finally demonstrating through little pharma at first, products approved by FDA based on the targeting approaches. And I think this slide here shows the targeted therapeutics that have been approved by FDA through controlled trials. But many of these trials, if you show a good relationship with the mechanism and the response, aren't your traditional long 10-year trials, 1,500 patient trials with overall survival assessment. These approvals can happen within 3 years. So you do a phase 1 study, and then you go right to a proof of principle trial. And if you show that the mechanism is correlated with a high response rate, the response rate that FDA has approved products for, we'll get into this a little bit later, is above 40%. That's almost automatic now. But if you show evidence of response rates as high as 30% of patients, then you're probably got a good proof of principle in a small number of patients. And that's the basis of our MSI program. And so I'll go over that. But if you look at programs that now have altered their cancer strategy, and you know, in standard of care, you're not going to generally say we're going to just do target therapy, not chemotherapy. There aren't many target therapies out there still. We have probably more than anyone in the country right now. But if you look at what are the results of all my patients are treated with targeted therapies, just in general, looking back 5 years, and what are the results of all my patients getting, you know, non-targeted therapies, chemotherapy? And if you do that analysis, a team of centers looked at this in thousands of patients, and they found that the survival of their targeted therapeutics was a year longer, better than a year longer than those that weren't. Well, that's something. But guess what? The basis of that wasn't the therapy you necessarily went on. It was the thought your clinical management team had on how to treat you. And that's where we need to be responsible. You know, we know this now. So if we have a patient, we know what their gene mutation is, and we decide, well, let's just put them on this study. If we don't have a study, then okay, there's not much we can do. You try what you can. But if you do have this study, and you do have the technology to understand what the signal is, I don't see why we wouldn't do that in every patient. And that's when we went to, we have to do the molecular profiling in every patient. And if we have a therapy that relates to that signal, then let's get them on. And at the very least, we should be able to, in the overall scheme of things, buy those patients an extra year of survival. You know, and that would be a worst case scenario. The newer stuff coming out is more active than the old stuff. So, this slide is in here because there's EMD Serrano has a PI3 kinase inhibitor that they'd like us to get engaged in the MSI program with, and we're probably going to move this forward. As you can see, it's a pretty broad opportunity across multiple cancer types. But the other reason I put this in here is that when you have a mutation and you target that mutation with your therapy, that's a good spot to target. But if there are other mutations that are upstream from it, and it all goes through that hit point of your therapy, all of those are going to be affected. So what we're asking companies to do is to assess the bioinformatics of their signaling target, where their therapy is blocking, and what's above it that could all go wrong. So if we have a patient and they have the mutation of right PI3 kinase, okay, great, they're a candidate. But there are other genes that are upstream from this, and if we can know what those are that go through that, then they too should be sensitive patients, you know what I mean? And so we're trying to understand what that is, and that's what EMD Serrano has agreed to do. There's about, I think, 8, 9 signals that make sense. Now some, when they get above, they don't all go through, the further you are from that target point, there are other signals that they go through. So it may be where it's only partial. And EGFR is partial with RAS. There is some activity outside of RAS. It doesn't always go through everything RAS, so it still may have some activity. But that's why knowing the signals and how common it is in different cancers is valuable. This is our profile as of, I don't know, in the last couple of weeks. And you can see the number of patients with the signals, and you can see the living patients. The reason we have the living patients is the patients are signing consent now, so we can call them up if we identify a signal that relates to a trial that we have or that we're bringing in. And so we're going to expand this program so we can bring these patients in more aggressively by calling them up and tell them about the studies. So, so that shows how we'll be operating the program as you go along. This just shows the aspect of the MSI program. And bottom line is, statistically, if you treat 24 patients and you have 6 responses, that would put you in position as achieving the minimum statistical significance against an expected response rate of less than 10%. Typically, when a patient comes to our clinic, they failed frontline and second-line therapy, oftentimes third-line. There's no standard of care, and typically, to standard of care or to any chemotherapy, there's less than 10% chance of response. So if you look at no activity would be less than 10% chance of response. If you see 30% response, then that suggests that the technology you're giving them has activity. And if you have that, the point is you may be able to power up a study probably double the size of it from 30 patients to 60 patients, and that situation, call it a rare orphan opportunity. The rare orphan is the tumor type plus the gene signal. That's less than 200,000 patients. That's what rare orphan means. And the FDA worked to get a fast track approval. So that's what we're trying to do, get this stuff out to more people. So does that make sense? Does that make sense to you?
Oh, sure. I'm like 10 steps below Rachel.
No, you're not.
No, you're not.
You are not. We're going to prove that to everyone. Next time. Deal with this time.
We're all in the same place.
Yeah, I know. Me too. Okay, so here's why everyone doesn't do it. When you get a product approved from market, your package insert tells you where your target population is, and the insurers are basically guided to cover those payments on that target population. Well, when you target, let's just say RAS mutation of lung cancer, well, that's 30% of lung cancer patients. So technically, the way some of these business guys at pharmaceuticals look at it is, well, wait a minute, why did they develop a therapy to not allow me to have 100% of lung cancer patients? So some companies actually know they're going to get 30% RAS mutations in a trial. You can calculate how big does the trial have to be if 30% of it is RAS mutation, so you can get the approval in lung cancer rather than the sensitive group lung cancer. So that goes on today. FDA is aware of it. So I've been able to work with FDA behind the scenes to basically help highlight that negative aspect. And so they've worked good with us on a lot of trials to move things forward, and that's positive, you know. But it's up to us to try to prove is what we're saying right. Do you really get better activity when you target the target? Is that really true? I mean, in mice models, it sure as heck is, but you know, we got to prove it. People, cancer, after they've been through a bunch of therapies, there's other reasons why people don't respond to treatment. Does the delivery of the treatment getting the tumors, that actually work? You may have a great thing that knocks down the gene signal, but if you can't get into all the tumor cells, then it may not have anything to do with the matching. It may be that the delivery is a problem, you know. So there's a lot of reasons to sort out. Ideally, 100% of it should work. And so there's a 30% other issues if a product that is considered a good product. You know, that's why that 30% number. So the other problem is that it's hard to get molecular diagnostics. As you guys know, it's a bunch of pages long. It's impossible. Here's the gene mutations. What the heck is that gene? I mean, what do people do with it? And then if you do know what that is, well then, what therapy can you use to act on that? Very few offices have therapies for targeted approaches. What I showed on the FDA-approved batch of agents right here, that's the best any office will have. You can order any of these things from, you know, from McKesson or whatever. But that's not many. So most of the signals you get aren't going to allow an oncologist to do anything with it. It allows us a little bit more, but we still don't have most of the signals. If you look at all these ratios, if you look at all the gene signals, we don't have therapies for the majority of these. So try to figure out what pathway to along, where it might be close to, and try to get more of these therapies into us. That's basically what we're doing. But you can see why people still, it's hard to get excited if they don't have the targeted therapies. So, and that's basically the main reasons. So the way Ewing's comes into it is, if you think about all the molecular signals that are out there, there's several thousand that relate to those 12 signal pathways that affect cell growth. And so of those several thousand, you'd think a therapy to block any one of those should be working, but all those 12 functions have, you know, because they're critical functions for the body and a cell to survive, they have pathways that protect a breakdown of one signal pathway. And so so sometimes it won't be 100% effective because there'll be aberrant pathways around it. So, but one gene signal that is very obviously related to stimulating cancer growth are the fusion genes. That's where Ewing sarcoma came into play. It's a fusion of two normal genes, EWS, FLI, and EWS makes one protein and has one structure like this. FLI has another protein, another structure like this. It didn't go all the way, so it's like this. See, notice that I did not do that. So, because it was right point towards you, Rachel. So, I wanted to make sure that I didn't do that. Everyone's a witness. So
you wanted to, I know.
I was there. different proteins, okay? Well, neither of these proteins will act to turn on cancer growth, okay? FLI and E2S by themselves have something to do with nerve function and red cell function, actually. But if you get them to fuse at a certain point, it'll form a protein like this. And this protein will bind to a cancer growth stimulating area. It's a transcription factor. And when it binds to that, it'll stimulate cancer growth. Well, that's well proven. 100% of patients who have this in their cells are going to have cancer that contains that gene signal. So it's a logical area to go after and build a knockdown to. Can you prevent that signal from making its protein? And if you can, does that take away the cancer? So that's what was conceived with the Ewing's approach. And shown here, the traditional way of going through that is to take cells with Ewing sarcoma, throw your agent against it who's supposed to block that, and see if it harms those cells in the test tube. That's called in vitro. Then you take those cells, you inject them into mice, and you let them live for a little while in mice, and you figure out, before you do the study with your therapeutic, how long will they live if they've just injected the cells. And most of the times, these mice will live an average of about 35 days. They're all dead in 35 days, okay, right here. The black line is the mice. Black. Do you wear black?
Dead.
But that's good. That's bad. That's the mice. If it's cancer, it's good. Mice is bad. So, anyway, the point is, you know what your model's showing you. So now when you give them the therapy that's supposed to work on E2S FLI, can you improve the survival of those mice? And so what was done with the BioSH E2S product, and this is just to show you the example, the green line are the mice that got the high dose of that. And they did well. They still had some limits as you go further out, but part of the limits is because the dosing was stopped after a while. So in people, we continue the dosing. So that's encouraging. It was statistically significant, but for FDA and for our own comfort, was it really related to the E2S FLI knockdown? So then you go and you biopsy the tumor, and that's why you see patients that we have get biopsies, and you try to show, was the E2S FLI high in these patients? And if you look on the curve on the right, the FLI is the thing that was measured here as part of the E2S FLI. It was very high before they got treatment, but then after they got treatment that was the higher dose, and at the low dose, there's a little bit more in the low dose than the high dose. It looked like it was correlated with a more knockdown with the higher dose. That's about 90% knockdown, maybe 93%. So it shows that the active ingredient that was causing the cancer basically was taken away. Cells died, mice lived. So that's what we're trying to achieve. Then you got to balance, and that's what is justifying going to clinical trial. So in Tyler's case, he was the first patient to get that therapy. And the best we've got on him, he's got stable disease. He had modest improvement in his lung function, and we're tracking to see where he goes with it. So he's continuing to get the E2S FLI. Because FLI knockdown has some control on erythropoiesis stimulation, we checked when his blood count was coming down a little bit for a period of time, we checked that. It turns out his erythropoietin level was fine, so it's not harming it. This is designed to not harm that purposely, but there are 4 nucleotides that connect to the erythropoietin that turn on. But anyways, it wasn't. So, we're tracking, we'll see what happens. So that's signal targeting E2S FLI. Does that make sense? Any questions about that? Grace, anything on your side? Right, cool. So then, the 2 part of the 1-2 punch is the immune function. And this is where I've had a lot of time. I mean, I really had a good friend of mine, Dale Ando, who was an expert in retroviral technology. When we set up the program originally, we brought in a immunotherapy where you inject a retrovirus containing a gamma interferon gene. Gamma interferon is the most potent substance that our immune system produces, that when it's produced next to cancer cells, it'll kill the cancer cells. Okay, so T cells that have, and then we all do. Everyone's T cells will produce gamma interferon when their receptor binds to a marker, and at that binding, that interaction says kill, and the kill product of those cells is gamma interferon. So all of our T cells do that. And it'll do it to viruses. It'll do it to bacteria. It'll do it to different kinds of things if it's armed with the right receptor. And so all of our bodies have such a ability to have receptors fit every single possible concoction that when cancer first starts coming out in the body, these cells rise to it, and they generally are killing the cancer, getting rid of it. But eventually, you start to run out of those cells. And some of these T cells last 40 years. So when you eliminate the whole population, you're kind of now giving an opening for the cancer to start to grow up more. And so if the cancer can eventually survive the T cell reaction or hide from it for a period of time, and it doesn't build up until the cancer gets to be so violent, trillions of cancer cells versus maybe hundreds of these T cells. The T cells may not have time to grow up. When they go in, they all get used up, and suddenly there's no more of these T cells left. So there's different ways that process gets a lot more complicated than that. But point is that the immune system can be completely depleted. So one of the strategies with immunotherapy was, can we replenish the immune system to be 30 years before, when you were keeping that cancer under control, before it got worn out, before it was exhausted, is some of the terms used. And so that's where the gamma interferon retroviral infection idea came through, and that's how Grover Cummings was managed. Were you around when Grover was first treated?
Not when he got treated, but shortly thereafter.
Yeah, it was right around then. And so, when the gamma interferon retrovirus was injected into his tumor, it created a reaction that crippled the tumor to the point where it couldn't protect itself, and the rest of his immune system evolved to now have the ability to attack the tumor, and all the disease in his body disappeared. And so he was our first dramatic case, and it never recurred in him. And so one of the things, the power of the immune system, once the immune system trips off, it can sort of self-potentiate itself to attack the cancer. So once you get it going again, it sometimes doesn't take repeat. That's why when you do an immuno approach, depending on what it is, sometimes you don't necessarily have to keep the doses going. Sometimes you create a new level of immune activity, and that'll persist and help throughout other therapies. Eventually, it gets exhausted again, but it's there. And so there were a whole series of dramatic responses. Jeannie's dad was one of the patients. We actually made tag to actually help him. It's kind of cool to be able to do that. And he had a good response. Connie West, who's on her IRB, that's her right in the middle there. And it's amazing. Remember they came and they thought she had recurrence? Yeah, we stopped them from giving chemo, and it's just all settled down and healed up. So she's still doing fine. She went to Africa again.
Yeah, we saw her a couple weeks ago for the 20th anniversary.
Yeah, that's cool. So something's going on. This wasn't just by accident, all these spontaneous remissions. So that's where, we got into trying to persist with developing immunotechnology for the part of the 1-2 punch. And but the immune microenvironment where the cancer cell lives is real complicated. Cancer cells produce tons of things to protect itself. The checkpoint inhibitors there, the Treg cells. It's very complicated. Bottom line is that it's really hard to sort that out, and you can have variable effects depending on what's more or less active there. It's hard to measure all these interactions, but at least we know some dominant things. Markers on the surface of cancer cells tell the immune system what they are. TGF-beta is your biggest inhibitor of all the immune system trying to attack cancer. GM-CSF is probably your best cry signal to call the immune system to the area, and gamma interferon is probably your best toxin, okay, to hurt tumor cells. Oh. So if you can focus immune response on those elements, you'll probably get some success. But one of the things that's been exciting, a few times it's happened where patients' tumor cells were put into a test tube to expand their T cells against the tumor cells. And in a few cases, patients have had dramatic responses where the cancers completely went away. And technology has since developed in the last couple of years where the tumor cells from that patient that were stored at the beginning could be identified based on their gene mutations, and specifically within the gene mutation, which peptide, which piece of that gene mutation was operating to be the neoantigen of the tumor cell. And it turns out these patients, there's only a few of them, they've had these complete dramatic responses to their own T cells taken out of their body, exposed, and back into the body. The neoantigens in these few patients have been identified from this very complicated analysis takes about a year to do. And so, but it shows how if you can train your immune system to attack the relevant tumor marker, you can indeed completely alter the cancer growth pattern. So now I can wonder, well, if you can do that on these two patients, we know exactly how it's done, why can't we do it on more?
Yeah.
Well, we can. It took about $200 million on each of those two patients. Right. So, okay.
That's a big problem.
That's a big problem. But it's like all technology. It always starts off as a big problem, but then it becomes a convenience, except for video games, kids. I don't know about that. But the point is, I'll get into that a little bit later on what innovative ways you can look at that. But what we do know is that the neoantigens are important. And there is a surrogate to neoantigens. That's what tumor mutation burden is. If you have a bunch of mutations, okay, you have a whole sequence of DNA and multiple pieces of DNA, and if there are a certain number of mutations per segment of DNA, that term is called tumor mutation burden. It's how many mutations occur along DNA with a certain length of DNA. I forget microbytes. I don't know what it is called. I should know that. I don't remember. Anyway, it's certainly the DNA. And so if it's high, it tends to suggest you're going to more likely have a higher proportion of those things on the surface somewhere, and you're likely going to have a higher neoantigen ratio, and hence you may have, by having more neoantigens, if you use an immune response and you activate your immune system to look at the cancer more closely, you're more likely to have a chance of generating an immune response against one of those neoantigens. And so that's when Margaret Brown came in, and we had seen these responses from other patients, Grover and Connie West and everyone, but those, they were on therapy, and wow, look what we got. With Margaret, it's like, wait a minute. Before we started on something, we had a choice of several things: a signal, chemo, immune. What's her tumor mutation burden? Does she have that signal for an immune sensitivity? And we don't know for sure if that's real or not. But we thought, well, she did. She had a high TMB. So, okay, let's put her in the development study. And lo and behold, this is her, and she had a nice dramatic response. You can see how much her tumor shrank. Oh, that's not Margaret. Next slide. Yeah, there she goes. And the whole time her cancer is being reduced. I think this is like a 67% reduction or something. It's a big reduction. And there's multiple lesions. This is just one of the main ones. She's out fishing.
Yeah.
I mean, she's getting treated while she's sleeping. She's getting treated while she's like playing with her kids, going to the zoo. So that's how treatment should work. And so now sometimes you don't have time. In her case, you did, but some patients come in with pain. It's growing fast. It's obstructing bronchus anyway. That's why you need to first get that signal, stop that signal, get that quick response. But there's multiple signals, so we need something else to hold it under control, and that's where the immune response come in. And this would be proof of principle of that. You know what I mean? So now I like it when I can tell a patient. I couldn't tell her for sure, but now the next TMB patient that's high, I'm going to have a lot more confidence, and if we can immune response to them, that they're likely going to have a better response because of that knowledge that we have on them. So then if you look at
Well, let me ask you about her. She had probably 13 or so mutations on Foundation Medicine.
Yeah.
Were these, the TMB, was that reflective of various ones, or is that predominantly one of those mutations?
No, it was non-specific. TMB is non-specific.
Okay.
We're probably going to be able to refine that a little bit better, because I think if there was a DNA repair mutation, there are certain mutations that would conceivably...
She had a DNA repair.
Yeah, that would conceivably be even more sensitivity. The world hasn't come out with that thought yet. That's just us, but we seem to be on target with most of these things. It's weird. Even though everyone hates us, but I don't know. We still keep helping patients. So, point is, at this point, the literature says it's non-specific, but I think there are relevant mutations like you're suggesting. But I'd still go by TMB as the main focus. But the TMB was validated in several studies. This is just the one that was a product approval trial with a checkpoint inhibitor, and in that trial, again, those patients with the high TMB did much better than those with the low TMB, both for tumor shrinkage and survival. So it's definitely a signal. This is one that Leah helped me on, but we identified 29 patients who had a 10, there it is, megabytes. I don't know what MB is. That's terrible. Well, 10 MBs of coding DNA. Anyways, their mutation burden was high of these 29 patients. I think 2 were unevaluable, and of the 27 others, 7 had gotten immunotherapy, and you can see them there, and 21 didn't get immunotherapy. They got signal, they got chemo, or something else. And so, of the 6 that got immunotherapy, before we did the analysis, in my world, if you have stable disease for more than 6 months, that's good enough. PR, CR by Reese's criteria is obviously good. So 6 out of 6 had responses. Whereas if you take the other 21, same TMB level, 1 out of 21 had a response. So I'm thinking we're probably on target with the TMB being a relevant factor. And then we talked to Foundation Medicine, they put it in their report. They believed us, they were getting evidence of the same thing. So that was one of the things that we did. As a program, we don't realize it, but worldwide we affected. Now, we're just sitting here talking about it, but thousands of people with high TMB, people who realize this on the Foundation report and all that stuff, they're being treated with, molecular, with immune therapy. And a lot of it was because of us. We're not going to get credit for it. God knows. So we're kind of, dusting off our little wings and stuff, hopefully, But point is, it's a good thing to do to help patients. And that's the idea. So, so then, are the unique criteria on the immune system that are outside of TMB? Well, guess what? These fusion proteins, they're actually expressed on the surface of cancer cells as well. So not only are they growth stimulators, but they can be detected by the immune system on the outside of the cancer. So if you go back into Ewing Sarcoma, it turns out responses can be generated in Ewing Sarcoma. This is some nice stuff showing tumor shrinkage in Carly and Andrew, 2 patients, and the LA spot correlation with the responses. And with recurrent disease, the LA spot coming down, and so it just suggests that there indeed is immune activity, and this is how it operates in the body. And others have done work showing dendritic cells attacking EDOS-FLI antigens. So bottom line is it all comes together, and you can actually measure circulating EDOS-FLI DNA. And the measurement of that DNA, if you look at this slide closely, I won't go into it, correlates with tumor shrinkage to chemotherapy, to any therapy. Point is, if the Ewing sarcoma mass decreases, the EDOS-FLI will decrease, and if it increases, it'll increase. And that was shown in animals and in people getting treatment. So we're trying to get that for our patients to get measured. So I think that's probably, I should save new antigens for later.
Poor Tasha.
Tasha can't get further. So, that's good enough. So anyways, why don't we stop there? I think you get the idea of the 1-2 punch. The idea then is with any cancer patient has DNA mutations, they have a minimum of 3, there's likely more. So if we can find out what the relevant ones are, and any cancer cell has unique markers on its surface, if we can find what those unique markers are, there's no reason why any patient can't have this 1-2 punch approach towards that. You just got to get the tools out there. You got to get the right molecular signal targeting therapy, the right immune targeting therapy. So
would that change the way we treat cancer overall? I mean, that'd be a better approach, obviously, it sounds like it.
Yeah, it'd be
with better response and less toxic, right?
That's exactly it.
So we would start seeing that with all kinds of cancers and treatment centers across. If that was
exactly. Yep. The obstacles on that is that it's a smaller population. So typically when a company develops, there's a lot of companies that only have 3 or 4 cancer products, but they generate billions of dollars. Well, if they only had 3 or 4 of these molecular targeting products, their user population is going to be much smaller. Even though they're getting these great responses, they're going to be much smaller. Additionally, in that smaller population, the response is going to be better. So are they going to get repeat treatment for a longer time period? Probably not. So what we're doing is we're creating, from the business side, a market that's much more difficult to make profit off of.
You're putting people out of business.
Right.
The companies that are doing it are putting themselves out of business.
Right, right. So then the focus is, you focus on the revenues you can create and sort of fake, I guess I hate to use the term fake news, fake therapy, but almost to some extent in a targeted therapy that's used in non-targeted patients, like the original EGF TKI, it was kind of like fake therapy for the majority of patients, the 75% who didn't have the EGFR mutation. Now, you still get some response, which I guess not terrible, but you just, our position is to not develop that kind of technology, or to develop it in the matched population, the right population. So that's what we're trying to do. At early stages, you sometimes don't know, but at least you can try to be mindful of that. And there's got to be ways, if you have a better therapy, of even allowing the big guys to make a bunch of money off of it, to still allow them to make a bunch of money. I mean, they could theoretically charge more for a shorter period of time, They can create programs where instead of doing a 1,500 patient trial to get the product approved, which would take 10 to 15 years, they could do a 60 patient trial that got Keytruda approved for the patients that had a 50% PD-L1 expression level, 61 patients. So now, wait a minute, if we can think ahead and bring 5 products in and start 5 60-patient trials at once, well, we could get 5 approvals over the same time, and that's only on 300 patients rather than 1,500. So it's doable. It's just that they have to do it. And what it takes is being intelligent on what you're doing. You got to have the 5 right products. Now, if they're matching the signal with the target, the old days of cancer, you have to do 10 products to phase 1 to get one of them approved. Well, I think if you have the right matching, you can prove the mechanism. This should work, I would think, 3 out of 4 times. So, but their model is, well, if we want to get this equivalent population, we're going to have to develop 5 or 10 products. Well, if only 1 out of 10 works, then we have to develop 30. That's going to be too expensive. Can't do it. So that's the way they're thinking. They don't get it, unfortunately.
Wow.
So that's the obstacle right now. Huge fight.
And the FDA is...
The FDA wants to target therapy by far. Yeah. And life's easier for them.
So
Yeah.
who's stopping? Is it the drug companies? Is it the politicians?
Oh, everyone, and so our little baby program isn't under the pressures of, MD Anderson and stuff has to do it. And it's not like people are consciously doing it most of the time. That's why I'm explaining it to you. Most people don't know. Even the business guys, the company don't really know. They think it's still 1 out of 10, But I think as our program can enlighten other people, the best way we can do it isn't by complaining about it or talking about it or saying, oh man, how dumb they are or whatever. That's silly. It's to just get the products approved, get the results, and let people, how did that happen? So that's what we're trying to do. So hopefully the next 2-3 years we'll pull it off, and that's what the MSI program is aimed to do. So that makes sense. Okay. Well, that's all then.
Thank you.
Thank
Awesome.
you. Thank you again, Dr. John.
Is there any way to make, I don't know, this sounds weird, to increase tumor mutation burden in order to make a drug work that wasn't working before?
Yeah, you definitely could, but each mutation that you create could have an added resistance factor and added this, so it'll probably balance itself out. The other direction would be the tumor mutation burden is a surrogate of the actual neoantigen. We define the neoantigen. And so, just since you asked that, I don't know if this will come out. This is pretty cool. There are 2 variables. There might be a different way of finding neoantigen where you can do it for a couple $1,000 rather than a couple $100 million.
Wow.
And it would be in a 5-day process rather than a 2-year or a year-plus process. So there's a company called Terrace. They're in Dallas, actually. I didn't even know it when we started working with them. and they developed these aptamers. These are little tiny molecules that are sort of like antibodies because they'll bind to little niches, little peptides all over the cell. And they found that if you can put a cell in a test tube and put 10 to the 7th of these aptamers around the cell, you'll get about half of them to bind all over the cell. But half the cell will still have some exposed areas that aren't binding these aptamers. So then if you put 10 to the 15th aptamers in that test tube with the cell, all these aptamers, each one is a different size and shape, different length, and different binding niche. The entire cell will be coated by all the aptamers. So then they're stuck with that and wondering, what do we do with this? So then when I heard them talking about how they're frustrated with that program, we were talking about other stuff. They do RNA expression analysis and stuff. I said, well, wait a minute. If you've got these aptamers that do this, and if they get kicked off an area, can you collect those aptamers? Oh, yeah, sure. Subdivided, whatever it is. But the point is, if it gets kicked off an area, they can using mass spectrometry, they can tell us what that peptide sequence is, and then if they know the peptide sequence, they'll know what the protein is. If they know the protein, it should match up with the gene mutation. So now the question is, well, how do you know where to kick the aptamers off? So I told them, well, we've got these T cells that are generating these positive ELISpot response. We've got 8,000 samples of T cells from 400 different patients. And when they bind to the tumor cells, they release gamma interferon. And the only mechanism known today of T cell binding to tumor cells is through what's called the TCR neoantigen interaction. If you don't have a TCR, which is on the T cell, binding to the neoantigen on the tumor cell, you won't release gamma interferon. So these release gamma interferon. So now we know that we have a vehicle that will bind only where the neoantigen is. So that neoantigen, if it's coated with aptamers, when the T cell comes along, the question is, will the T cell compete with the neoantigen and kick the neoantigen out? Will the T cell be able to bind better than the neoantigen? Typically, you would think it would. Typically, when there's a biologic purpose for a binding versus a synthetic constructed that's coating it, typically you get competitive advantage to your natural vehicle. So we don't know yet. We're working out to test that. And point is, if you take the aptamer-coated tumor cells, you throw the T cells in with them that are known to turn on the ELISpot response, and you collect those aptamers, we should be able to tell what the peptides are. Now, there may be some mixed responses, so we want to see if those aptamers that are kicked out match to any known mutated DNA signal. And if we get that, we're going to be thinking that's probably it. And if we know what that is, we can basically then take those peptides or the proteins themselves and take them in culture, expose the patient's own lymphocytes to those, and grow them up to ungodly numbers, and just give them infusions and just keep going until the tumor goes away. So that's something that was conceived. I guess I had the idea of the competitive binding, but Neil and Donald and the kerosene, we all worked on that together, and so that's something that's going on behind the scenes, to see what happens. But if it turns out to be real, it could be pretty good. So, yeah.
Also, you think we can try it out?
Well, the next thing I wanted to do, I wanted to give them the first samples. They're getting ready to run it. The first samples I thought would be most appropriate would be, we have 80 patients who went on the phase 1 trial that got vigil. You could do it with other things that have to be vigil, but I happen to have that. So of those 80 patients, 48, they all had failed standard of care. They're all phase 1 trial patients. All came looking for phase 1 trial, and they happen to qualify for that phase 1 trial. And so 48 of them are alive after 2 years. And I think 30 of those 48 are still alive today, which is about 3 to 5 years. And so what I wanted to do was take samples that we've already got storage in those patients, let them run those patients. They find the neoantigen in those patients. Most of those patients, we're going to do a long-term follow-up. We're pulling the records. We're just trying to get the stuff sorted out. But once we get the long-term follow-up, we'll find out what else they went on. Do they still have cancer? Or do these patients, I don't even know, we haven't found, maybe all these 48 had their cancer disappear for all I know. But I expect most of them still have their cancer, but it's just stabilized or it's slowly growing because the immune system's still there trying to wear it down. And so in that scenario, if they still have the cancer, and I find the neoantigen to that patient, what I'd want to do is expand that patient's T cells and take that big pool of T cells and give it back to that patient and see if we can get a shrinkage to that patient. If we do, that validates the activity in the clinical setting. You see how that would work? So we're sort of just going through politics of setting up the contracts and all that stuff right now. Some internal stuff actually even. But the point is, you can see where this stuff can go. So we can get these ideas, but we actually have the ability to kind of carry those ideas out. You know, anyone can come up with an idea, but it's based on what we see with our patients. Can't do more. Remember the CDKN2A? I have no idea why Takeda wouldn't want to pursue that. Ridiculous. But you know, it seemed like that target would have been great for their product, but they just didn't want to do it. So so you can't always get them going, but you know, the more we can, the better, I think. So does that make sense? Yeah.
Give us a